thanks the organizer for giving us the opportunity to share our work, and thanks also to the many funding agencies and many disinterested collaborators for making it possible. Non-alcoholic fatty liver disease is an important health burden for the health system. It has a complex and long-term evolution from simple fat accumulation in the liver parenchyma to the potentially progressive type called non-alcoholic steatohepatitis or NASH, which evolved in 5% of the patients to NASH fibrosis. It's important to do a proper diagnosis of patients because they could have many different prognoses. In fact, although NAFLD patients suffer from 50 times more cardiovascular disease complication, in those who develop advanced fibrosis, the progression is fast and the mortality is high. Neutral lipid accumulation, like triacylglycerols, is the marker of the disease, but we will talk here about less abundant lipid species like ceramines, dihydroceramines, and dihydrosphingolipids in general. These ceramines are synthesized from condensation of serine and palmitic acid, which is also a precursor of triacylglycerols. In insulin-resistant prediabetic states, there's an oversupply of fatty acids from adipose tissue to the peripheral organs, which synthesize triacylglycerols. But by a mechanism of spillover, it also synthesizes ceramines, which enhance the capacity of the liver to synthesize even more neutral lipids. Blocking dihydroceramine synthesis using, for example, inhibitors of the delta-4 desaturase or in knockout animals protect from development of an accumulation of fat in the liver. But at the same time, they accumulate a huge amount of dihydrosphingolipids in the liver. This is somehow paradoxical because what suggests two recently published studies, for example, is that dihydrosphingolipids are increased in the liver of NAFLD patients and that ceramines do not change. We believe that a better understanding about the role of this kind of dihydrosphingolipid species and sphingolipids in general is necessary in connection with the progression of the histological staging of the disease is necessary to draw conclusions. Histologically, the classification of a simple steatosis or NAFLD from steatohepatitis is based on the presence of several features with different prognostic and severity value. Steatosis is diagnosed by the evidence of fat infiltration in the liver parenchyma. The inflammatory stage is grade according to the infiltration of inflammatory cells or the presence of, or by the presence of ballooned hepatocytes. And it's grade together in the activity score. The intersection between the two scores defines the diagnosis of NASH. Fibrosis is evaluated separately by the evidence presence of thin threads of collagen fibers to thick bands which are observed in cirrhosis. A cutoff value of stage 2 is set to define the difference between NASH without fibrosis and NASH with fibrosis. This is clinically relevant because the presence of NASH fibrosis is the fundamental predictor of liver transplantation in patients. The aim of this work is to study the relationship between dihydrosphingolipids and histological grading and staging in NAFLD. To address this question, we made a comparative study between animals and human samples. First, we developed an animal model of high-fat diet, which was a diet-induced obesity model, which was fed a high-fat diet enriched in cholesterol and sugar in the drinking water, and we fed the animals up to 14 weeks. The same set of animals, or a different set of animals, were injected with carbon tetrachloride for 10 weeks to induce a more advanced fibrosis stage. You can see here that animals were obese in comparison with their control litter mates, but addition of carbon tetrachloride reduced the weight of the animals. We sacrificed the animals at different time points in order to have a representative samples of all the different stages of the disease. Our patient cohort consisted on 200 NAFLD patients, which were diagnosed by a surgical biopsy or a percutaneous biopsy. Most of them suffer from related metabolic complications like type 2 diabetes, high blood pressure, or dyslipidemia. But above all, more than 80% of them suffer some degree of obesity. Liver of animal and humans were classified according to the SAF classification algorithm. Our methodology, we measure the liver and plasma tissue with using a targeted lipidomic approach, which was validated in the publication above. The sample were processed in different analytical batches. On every batch, we set a long-term calibrator and also 2-3 levels of quality controls to check the analytical process. You can see here that, for example, the overall behavior of our sample over a period of 1 year was quite good, and that even in the worst case scenario of the quality control high, the coefficient of variability was 20% for the lipid species above and 30% for one species of triacylglycerol. Okay. When we fed the animals with a half-fat diet, you can see that the weight of the livers and also the weight of the animal increased, as also increased the accumulation of fat in the liver. The animals were insulin resistant. It was evidenced by an oral glucose tolerance test. Microsteatosis developed early at 22 weeks and evolved into macrosteatosis at 30 weeks. Inflammation was evident at 30 weeks and evolved quickly at 30 weeks. It was manifest by the presence of a cluster of inflammatory cells, crown-like structures, and balloon hepatocytes. This happened at the same time as a steep elevation of transaminases. Fibrosis develops at 30 weeks from stage 1 perisinusoidal fibrosis to stage 2 perisinusoidal and periportal fibrosis. The addition of carbon tetrachloride reduced both the body weight and the liver size of the animal, but accelerates the formation of fibrosis in the animals, who progress from stage 2 to stage 3 rigid fibrosis with an increase in the necroinflammatory damage. We check our liver findings by protein expression of several markers related to the lipid accumulation, like perilipin, stellate cell activation, like alpha smooth muscle actin, or fibrosis collagen. But we also can see that there is variability in the results found in the liver for the expression of these markers. That's one of the reasons to classify them according to the histological findings. And as you can see, for example, here, the mice which were classified as having NASH have a higher steatosis score than that who have only simple steatosis. But when fibrosis increased, there was a decrease in the concentration in both scores, the steatosis score and the activity score. We then performed the lipidomics analysis, and you can see that overall the mice which were control mice have lower triacylglycerol and in general neutral lipids than the one who had advanced fibrosis stages. We also found an elevation of acylceramines, a class of sphingolipid which is derived from the condensation of fatty acid to the free hydroxyl group of ceramines. We also observed that the behavior of triacylglycerols and ceramines were somehow different in the NASH fibrosis group. And in fact, if cluster analysis separates quite reasonably the higher fibrosis group who had lower triacylglycerol levels from the lower fibrosis group who had higher triacylglycerol levels. Our conclusion here is that in advanced fibrosis, is that when steatosis increases, it also neutral lipid, there is an increase in neutral lipids, but when in stages of advanced fibrosis, the neutral lipids decrease, but the dihydrosphingolipids still increase. This is also supported by the result obtained when we did expression of several enzymes which are related to the the genes, when we follow the gene expression of genes which belong to the pathway of ceramines, most likely because of an stimulation promoted by fatty acid overload. Then we wanted to know if these findings were somehow extrapolated to human NAFLD. What we did first is to compare the histology of a patient with that of our mice model. And you can see that the trends observed in the histological score were quite similar. The main difference is the scoring of the activity, which is somehow overestimated, and this may be because of that hepatocyte ballooning is difficult to differentiate from hypertrophied hepatocytes in the liver of mice. But even though the behavior of transaminase elevation in NAFLD patients and in our animal model was quite similar. Then we performed lipidomics in our human liver samples, and you can observe that the trend of neutral lipid accumulation were increased as the disease progressed. you can show, for example, you can see that in these triacylglycerol species, acylceramide species, and also dihydroceramines. We can compare. This is a comparison. Even in concentration terms, the results were quite similar. But unfortunately, we don't have enough samples from advanced fibrosis patients, liver samples, because most of our patients came from the bariatric surgery procedures and don't have these advanced stages. Luckily for us, we found a good correlation between plasma and the dihydrosphingolipid accumulation in the liver. It's interesting to note that we don't see a correlation between plasma, between the concentration of sphingolipids like ceramines, exosylceramines, and sphingomyelin in the plasma and those of the liver. Maybe because these kind of lipids are not very well tolerated by the liver. The correlation between plasma and a liver sample encouraged us to analyze the whole patient cohort in plasma. And you can see that the trend overall, it's similar. It increases the concentration in NAFLD patient, but again, you see that there is a difference between the patients who had advanced fibrosis in the NASH fibrosis group. And again, cluster analysis separate reasonably well those who had advanced fibrosis and lower triacylglycerol to those who had less advanced fibrosis stages. Our final conclusion here are that dihydrosphingolipids and neutral lipids accumulate in the liver of NAFLD and correlate with the scoring of steatosis both in mice and humans. And the appearance of significant NASH fibrosis reduced the concentration of neutral lipids, but not of dihydrosphingolipids in the liver of mice. And I will be happy to take any question you may have.
Thank you for the nice talk. Are there any questions? Okay.
Thank you very much for sharing your interesting findings with us. I am interested in your mouse model a little bit, and there the subjection to the tetrachloromethane, because as far as I understand, this is hepatotoxic, and therefore I'm wondering what the physiological significance might be, or why you think that this is maybe comparable to the way that these fibrotic lesions are forming in humans, maybe. Thank you.
Yeah, you're right. The thing is that it's very difficult in the mouse to produce a high advanced stage of fibrosis. And to accelerate that, you have to add something. If not, you have to feed the animals for, I don't know how much time. So I agree with you that adding a toxic agent might be the reason for some of the changes we observe. You're right.
If I understand you correctly, in a mouse model, you've seen that cholesterol content more than doubled. Is that quite correct? Or
cholesterol ester, you say?
No, cholesterol itself.
No, the free cholesterol, we don't see changes in lipidomics. We don't see many changes in free cholesterol.
This was on the graph.
Sorry?
It was on the diagram. As kind of...
Yes, it was. Free cholesterol was in the data, but it was only in one. I didn't comment it, but
right, okay,
we measure it. I don't know. Mainly, most of the cholesterol who came from that we gave to the animals was transformed into cholesterol esters.
Okay, further questions? If not, thank you again for the nice talk. And now we have a lunch break. And again, thank you all the speakers of this session.